Sorry. Okay. Blood sucking insects transmit. We have Plasmodium species. We have Wuchereria bancrofti, Onchocerca volvulus, Leishmania species, Babesia species. Then we have housefly, which is referred to as mechanical carrier, can be a source of entamoeba histolytica, which is E. histolytica. The dog is a source of Echinococcus granulosus and Toxocara canis visceral larval migrans. Then we have cat, is a source of Toxoplasma gondii. Man himself or human being is a source of Entamoeba histolytica, Enterobus vermicularis, Hemilolepis nana. And we can also have auto-infection, automatic infection. It may occur with Enterobus vermicularis, Strongyloides stauchoralis, leading to hyperinfection. So the infection is taking place within the body of the host. Portal of entry into the body. That's the point of entry of parasite into body. We have mouth. The most commonest portal entry of parasite is oral, by contaminated food, water, soiled fingers. That is, we have fecal oral route. Examples, we have Entamoeba histolytica, Giardia lamilia, may also be transmitted by anal, oral, sexual practices amongst homosexuals as well as lesbians. Then we have kissing, Entamoeba gingivalis, which is a normal flora in the mouth, is transmitted from person to person that practice kissing, or from contaminated drinking utensils. We have congenital infection with Toxoplasma gondii. We have Plasmodium species, HIV, etc. may be transmitted from mother to fetus transplacentally, that is through the placenta to the unborn child. Inhalation, we can inhale certain parasites. It's not only COVID, which is a virus we inhale. Airborne eggs of Lumbricoides, Ascaris lumbricoides, Enterobus vermicularis may be inhaled into posterior pharynx, leading to infection. We have iatrogenic infection, which means as a result of treatment, hospital treatment, a person can be infected. Malaria parasites may be transmitted by blood transfusion from donor with malaria containing asexual forms of erythrocytic schizogony. This is a stage of reproduction. This is known as trophozoite-induced malaria or transfusion malaria. Malaria parasite may also be transmitted by the use of contaminated syringes and needles amongst addicts, drug addicts. Well, another assignment that can help you be more acquainted with parasites is from head to toes of man are parasites, which means from the head to the toes of man, they are habitat of parasites. Using tabular form, write down the parasites that can be encountered. So parasites that you can encounter from the head, write the examples of parasites in the eyes, and so on. Then we have attributes of pathogenic parasites. Pathogenic parasites are parasites that cause diseases. A parasite can cause disease because of certain properties of pathogenicity. Here are some factors of those characteristics that parasites of pathogenicity possess. 1. Trauma. Hookworms, roundworms, teniaces, they cause relative slight physical damage as the filiform larvae enters the body. So there is traumatic damage of pulmonary capillaries leading to hemorrhage, bleeding at the lungs as these parasites penetrate the lungs. The affected area can result to bleeding. We have lytic necrosis. Amoebiasis is incriminated. Then we have entamoeba histolytical, which is also causative agent of amoebiasis, Giardia lamblia, and the rest. They secrete lytic enzymes which lyses tissues for its nutritional needs and help it to penetrate. So they break down, they have enzymes that break down tissues, and by doing that their nutrition is being produced. And we have inflammation. Examples of those diseases: trichinosis, leishmaniasis. Most of the parasite cause cellular proliferation and infiltration at the site of their location by fibrous encapsulation. So where the point of entering is, they can cause encapsulation at the surface of the skin. We have toxins. Amoebasis and Amoeba histolytica have shown to produce toxin-like substances that can affect victims. The toxins can be in the form of pus. We have allergic manifestations, visceral larval migrants, roundworm infections, the fluid encysted in hydatid cyst, and escape from cyst can cause fatal reactions. So the hydatid cyst is like a sac and it contains fluid, which are toxins. So if that cyst breaks open, the fluid comes out, the infected organism definitely will react, and some of the reactions can be deadly. Now, we have laboratory analysis, diagnosis technique that enable us to detect these parasites in samples. If we collect samples from the field, we bring them to the lab and subject them to certain demonstration, and we'll be able to detect our parasite. Demonstration of parasite through blood, stool, urine, genital specimens, cerebrospinal fluid, sputum, we have culture when we collect small number of parasites. To amplify the number, we refer to it as culture. Then we have skin test or skin snips to dictate microfilaria. We have immunodiagnosis, serological tests to dictate antibodies or antigens in patient serum. We collect blood sample and get the serum, and then we subject that to serological test, and that will be able to dictate our parasites from infected organisms. Then we have molecular biological methods. These include DNA probes and polymerized chain reaction, PCR, which has to do with molecular base analysis. To further help you in your studies, you have question that you should list 3 laboratory techniques used for diagnosis of parasitic infection.